Good morning, everybody. My name is Nagachandrika Pallam, Assistant Professor working in Sri Datta Institute of Pharmacy. Today topic is the Unit 5. The topic is Biowaviers and its criteria for the B.Pharmacy third year's first sem. So, introduction. So, what is Biowaviers? Here, Biowaviers is nothing but bio is nothing but the bio availability. So weavers is nothing but it is not considering. So bio availability not considering, the here not like that. What we are not considering means bio in vitro bio availability only we are considering. In vivo bio availability we are not considering. So what is the main definition concept is in vivo bioavailability and in vivo bioequivalence studies we are not considering. Only in vitro bioavailability, in vitro bioequivalence studies only we are considering. So that once again I'm telling that the main concept is simply we can say that only clinical trials we are not considering. In the clinical trials we are not considering, we directly the biopharmaceutical means the bio in vitro studies only we are considering. So in vivo bioequivalence and in vivo bioavailability is nothing but the clinical trials. So clinical trials we are not doing in the not conducting, that we call it as a bioweavers. So why we can say that instead of conducting expensive and time consuming in vitro studies, the dissolution studies should be adapted as a surrogative basis. Means if you are taking two formulation products that are which are equal, those why we have to conduct the again same test? So it is the conducting tests are clinical tests are very expensive. You know that clinical trials are very expensive, and also it is time taking. So to avoid this time taking and cost wise and time wise, we can reduce the we can reduce these studies in the meantime. We can go for the other products. So the main advantage means instead of means to reduce the expensiveness cost, we can reduce the cost and time consuming by these bioweaver procedures. So what are the bioavailability waivers means only APIs with the high solubility and high permeability, and which are formulated in a mainly solids. Okay, solids and IR formulations can be approved on this base, on the basis of bioweaver procedures. Means bioweaver procedures mainly consider, that means bioweaver procedures mainly follows the solid dosage forms, and also IR means immediate release oral formulations, that too APIs which are having the high solubility, high permeability, those drugs can follow these bioweaver procedures. Means to reduce the what is conclusion of this slide means bioweavers is nothing but we are not conducting the clinical trials. Whatever in lab we are conducting the test, we are comparing with the lab test and just we are submitting to the FDA. So FDA only suggesting this one. So we have to submit. If the product API having these qualities, we can negligible for the clinical trials. So what are the conditions there? In the guidelines we will see in next slide. Okay. Okay, so not only that, bioweavers, before going to this bioweaver type of the products, why this is happening means, if you are taking any product means, if you are same product, if you are studying again on the same subject, means same human trials you are conducting again also, you will not get same bioequivalent studies. Okay, so same clinical trials, the same response you will not get. So why we need to conduct these again these bioequivalent studies? So the risk of therapeutic inequivalence of the two immediate products can never be reduced to zero. Even if the full clinical studies is performed, also we will not get the same bioequivalent studies. That's why we will not go for this again clinical trials. That's why they are following the Bioweaver procedures. And conclusion of the comparative clinical studies and in vitro bioequivalence studies, in vitro bioequivalence test and Bioweavers are based on the statistics and the scientific data that consume the representative for the product at a issue. So that means they are how they are calculating this in vitro bioavailability compared with the in vivo means by the statistical data of the statistical and scientific data we are assuming to represent a product is a product. So the main Bioweaver guidelines is to reduce the risk of bioequivalence to an acceptable level. That is the main goal. So the pharmaceutical development works aim to reducing the probability of the manufacturing inequivalence formulations, taking into account of the critical aspects of the product is an issue. So that is the main concern. And in this mainly absorption phase you are taking. So why we are taking absorption phase? Because how much amount and how much amount of the drug available within the systemic circulation, how much speed it is available within the systemic circulation, that only decides the pharmacological action. So that's why mainly they are concerning about the absorption phase. So the test versus reference, we are comparing the absorption phase. So next one, what are the now we will discuss about the Bioweavers of the types of the products. So what are the different products we are considering means, solutions, IV products, and the immediate release products like low strength, very important, you have to consider the low strength drugs only. For the IR products we are converting into a Bioweaver procedures, and BCS class 1 and BCS class 2 are the questionable, and BCS class 3 also very questionable. So mainly what they are considering high solubility, high permeability drugs with the low strength. They are with the in a immediate release products, they are following this Bioweaver procedures. If they are proceeding for the extended release drugs means low strength drugs, they are considering. In topical doses form, what they are doing, what they are considering means solutions, aerosols, antifungal, those doses forms only they are considering. And inhalations, mainly inhalations and nasal doses form, they are considering the solutions. Okay, next one is here see this scope and advantages of the Bioweavers. So here these animals are protesting. See this figure. These animals are protesting, so there are no more experiment on us. That means we can, by this Bioweaver studies, we can avoid the animal test and animal clinical test and human clinical test by this Bioweaver procedure. That is the main concept of this Bioweaver procedures. So we can avoid this animal test, that is nothing but we are avoiding the clinical trials. That is only the main concept. Okay, so inside the product, what is the scope means, in scale up process it is very useful, and line extend. So if you want to extend the more more test, then we can consider these Bioweaver procedures and variations after the marketing authorization. Then also you can consider this Bioweaver procedures. And in between the procedures means when you are taking this a test, when you are comparing with the test with the reference product, that means when you are comparing with the branded versus generic product, these Bioweaver procedures are very useful. And what are the advantages of this Bioweavers means it is very, these clinical trials are very expensive, and sometimes these are unethical. Human rights, human ethical committee, sometimes they will object for the clinical trials. At that time, these Bioweaver procedures are very useful, and it also reducing the time, and also I already told you, same, it can reduce the time and also the production cost. Okay. Next one is limitations. Very, very important, these limitations. What are those limitations? Limitations means which are having the narrow therapeutic drugs. Those are not suitable for this Bioweaver procedures. And that means some drugs having the low bioavailability. So the therapeutic, the low means therapeutic range is very less. So that very less therapeutic range drugs only giving the best pharmacological action. Beyond that means what will happen? It will give the toxicities. So those drugs only need the narrow therapeutic range only. Some drugs should have the narrow therapeutic. The narrow therapeutic range only gives the best results. Otherwise, if you are taking beyond that, you are taking the therapeutic range of the same drugs, means it will give the toxicities. Those drugs are not suitable for the Bioweavers. Not only that, the BCS based on Bioweavers having the limited applications for the and the class 2 drugs. So why it is limited application? Class 2 drugs are very limitations, limited and not applicable for the class 3 drugs. So maximum they are not suitable for the class 2 drugs. Okay. And the dosage form meant for the absorption of the oral cavity, like some doses from these doses from like sublingual and buccal tablets are not suitable for this Bioweaver procedures. Okay. Not only that, the food effect of the food and absorption transports and effect transporters and route of eliminations were a very route of eliminations and overall drug absorption, the bioavailability, immediate release dosage forms, which are not considered as a biopharmaceutical classification. So before, if you first of all, what is BCS? That we will learn in next slide. Okay. BCS bio means Bioweavers mainly. I already told you, Bioweavers mainly concerning about the high solubility, high permeability drugs. Okay. So how it is classified according to the BCS classification? These are the class 1 drugs. So first of all, we will see what is the BCS is nothing but the biopharmaceutical classification system. So what are those? We will discuss in this slide. Okay. So class 1 is nothing but the very high solubility is very high, and permeability also very high. Those drugs we are we are considering as a class 1 drugs. See, in class 2 drugs is nothing but the low solubility and high permeability drugs, means solubility is very low, but it is easily penetrating into the systemic circulation, but the dissolution and solubility is very low. So those drugs considered as a class 2 drugs. You see, class 3 drugs is nothing but the high solubility, means the drug is solubilizing very easily, but the drug is not penetrating through the not observing through the small intestine, that those drugs we are consider as a class 3 drugs. Maximum class 2 drugs, class 3 drugs we are considered as a pharmaceutical, considering in a pharmaceutical preparations. But in research work, mostly class 2 drugs, class 3 drugs considering as a research work. Okay, then because if the solubility, low solubility means we can increase the solubility with the permeation enhancers, and if the permeability is low means we can add the permeability enhancers. So these drugs are very but in Biowaviers procedure, we should not add any solubility enhancers and permeation enhancers. We should not use. So it is not considering as a solubility enhancers and permeability enhancers. So simply we can say that class 1 drugs only suitable for this Bioweaver procedures, and class 4 is nothing but, already I told you, low solubility and low permeability. Okay, so in class sorry, in class 1 drugs, what are the considering in this class 1 drugs, the main rate limiting step is nothing but the dissolution. Dissolution is rate limiting step. So what is rate limiting step? So the product is, for example, if you are running very fast, at some point you are you are become very slow. That means that step we call it as a rate limiting step, and which factors are influencing that slow rate, that we call it as a rate limiting, the factors affecting the rate limiting step, that we will consider. So at which point the rate is slowing down, that step we call it as a rate limiting step. So here in class 1 drugs, where it is becoming very slow means dissolution is the rate limiting step, means it is solving very easily, easily solvable, but the dissolution is the main problem. So if the dissolution is rapid, then the gastric emptying will be the rate limiting step. So what are the drugs mainly example for this class 1 drugs means metoprolol, diltiazem, verapamil, propranolol are the example for these drugs. And class 2 drugs, what are the class 2 drugs means bioavailability of these products are limited by their solvation rate. These drugs having the high absorption and but the low dissolution number. These absorption class 2 drugs occur in the longer period of the time. So next one is the class 3. What are the class 3 drugs means? Here the absorption is limited. Here low permeability, that's why it is here the rate limiting step is permeability only. So absorption is limited by the permeation rate. So so what are those class 1? What are those drugs means? Cimetidine, acyclovir, and neomycin B, and captopril. These drugs considered under the class 2 drugs. Okay. Next one is class 4 drugs. So what are those class 4 drugs? These are not both the rate limiting steps are the both solubility and also the permeability, both are the rate limiting steps. So here what are the examples of this drug means chlorothiazides and taxol drugs. Very important, this classification is very important. Coming to this objectives of this BCS classification. So dissolution, if the drug is dissolution very fast means what will happen? solubility will be increased. Solubility also increased. Solubility increase means the permeability also increased. So the rate, what is the dissolution means? Dissolution means very rapid or rapid dissolution ensures that the high dissolution, it is not likely to be rate limiting step. So if the very solubility drugs, what is the rate limiting step means? Dissolution is only the rate limiting step. Okay, in a high solubility means solubility, high solubility to ensure the solubility is not like the limited dissolution, and therefore the absorption also. The high permeability, if the high dissolution means if the dissolution increase, the solubility also increases. Solubility increases, the permeability also increases. So so the high permeability to ensure that drug is completely absorbed during the limited transit time through the small intestine. So high permeability means it is directly absorbed through the systemic circulation, through the portal into the liver. So next one is data support for the request for the biowaviers is nothing but the high solubility definition, high permeability definitions, and also the dissolution, all those things. But we will directly go for the requirements of the BCS classification. So what are the requirements means? Study means a dissolution, very important. Dissolution test. So I'm not again repeating in dissolution test, so directly I'm explaining here only. So dissolution criteria, what are the tests we have to perform? So in dissolution medium, what we have to take, how much ml we have to take means 900 ml we have to take. Why the 900 ml only? Means the stomach contain 1 to 1.5 liters our stomach capacity. So 900 ml, so approximately we are taking the 900 ml. So our body temperature is 37 degrees. That's why we are maintaining in vitro studies also 37 degrees. So whatever the in vivo condition, our body conditions, same we are maintaining in outside also. That we call it as a in vitro studies. In vitro is nothing but the outside of the body. So same as it is like the body temperature and the body capacity we are maintaining outside of the with the instruments we are maintaining same temperature, same capacity with the dissolution test. So here we are taking our you already know that our stomach having the pH of 1.2. so the our gastric fluid, same we are maintaining it 0.1 n HCl, and pH power 4.5 and pH 6.8 for the stimulating intestinal fluid without the enzymes we are maintaining. And 12 samples we have to take. For each medium you have to take 12 samples. And this is the maximum we are using the pedal rotating. So how many rpms it is taking? Rpms means round per minute. So why, how many? 50 rpms. If the basket type means 100 rpms. So why we are taking the rpms means our inside the body we are having the peristaltic movement. That's why with the peristaltic movement the drug disintegrated under dissolution and solubility will be increased. So the same we are maintaining the outside of the means in vitro dissolution test also we are maintaining that peristaltic movements by maintaining the 550 rpms. Okay, the sample times, how to take this sample times means 10 15 20 minutes and 30 minutes, 45 minutes, 60 minutes we have to take, and we have to compare with the test and reference product must be similar to them three medias. So next one is additional considerations for this means the mainly excipients. What is the excipients means? BCS classification related to API without excipients. So first we have to take the without excipients. After that with excipients we have to take the take into a considerations. So however the literature, so if you want to study the excipient effect on this API, that also by this dissolution studies so we can perform. So next very Next one is the prodrugs. Prodrugs is nothing but the conversion site of the prodrugs to the drug must be considered. If it occurs before the intestinal absorption, then the permeability study of the drug must be done. In otherwise, the permeability study of the product must be done. So this is the considerations. So what are the next one is the Biowaviers procedures. According to the Biowaviers procedures, one minute. Sorry. According to the Biowaviers procedures, so according to the Biowaviers procedures, BCS class of API class 1 drugs are very suitable for this Biowavier procedures. So in this, what we have to maintain means the dissolution characteristics means very rapid dissolution drugs we are considering into a Biowavier procedures, and 3 different medias we have to take. And the first option is the very rapid dissolution and no further profile comparison we have to take. And excipients is most influence the absorption of the API, so that also we have to study. Already I discussed about this phentoin. They are changing this phentoin with the calcium phosphate to with the lactose. That lactose is having the high dissolution, means wetting property. Wetting property, what it leads, it increase the dissolution. At the time, what happened, the drug is more absorbed into the object in the systemic circulation. At the time, what happened, the more drug leads to more toxicities. So that's why we have to consider the excipients also. And therapeutic index means not containing the API with a narrow therapeutic index. Those drugs are not suitable. And formulations are the products. Product should be designed to be absorbed from the oral cavity only. Those only we are considering. So next one is the BCS, same like that. Okay, next one is the regulation provisions for the Biowaviers. These are the regulations of the US and UE means European Union and Japan. We are maintaining these qualities. Like we are in US, they are considering only class 1 drugs. In UE means European Union, they are considering class 2, class 3 drugs. Japan considering all the drugs, but US only considering class 1 drugs. Okay, and highly permeable drugs means 90%, below 90% they are not considering. Means it should be below 90 percentage, and means sorry, above 90% drugs they are considering. And here in UE, above 80% of the permeable means the drug bioavailability means permeation drugs they are considering. And Japan, they are not relevant. They are not particularly this percentage they are not considering. Okay, and rapid dissolution means dissolution should be more than 85% within 30 minutes. And these in UE means above 95% with the 15 minutes only. And these Japan, they are not requirement, so any drug they are considering. And media surfactant, the SUPAC is nothing but the scale up post approval drugs. So these are allowed to be if justified. And non in EU means they are strictly They are strictly discouraged, and in Japan means required if the low solubility, and intramural composition changes means soup package, all those things are not necessary, but it is very very important also. Okay, these are the surrogative properties. Next one is current pediatrics. So pedestals of the bioweavers. So what we are considering in BCS, what are the drugs we have to take consider means considering the dose solubility ratios and permeability dissolution behavior are very very important while considering the bioweaver procedures. And in vivo in vitro correlations, so what we have to take, the based on the correlation with the in vitro studies data into the in vivo profiles. So just why you are not considering in vivo studies means just we are comparing with the in vitro, then only we are correlating with the in vitro. That's why we are not considering the in vivo studies. That means clinical studies we are not conducting. Okay, CP is nothing but the composition proportionality source. So what we are taking means new products is qualitative the same, and quantitative the proportional of to the bio batch. So these are the consider uh, considerations and applications of bioavailability. Biowaviers means the main importance application is checking the variations of the drug products. So you are comparing the products. While comparing the products, these bioweaver procedures are very very useful. And when you are converting into a generic products, these bioweaver procedures are very very important. So what is the third application means the while developing a new drug products or bridging studies or pilot, when you are converting these pilot into scale up batches, these bioweaver procedures also. So in pilot is nothing but the R&D. When you are scale up means you have to increase the small volume to large volume. So when you are converting to a large volume, we are comparing with the pilot batches. So at the time it is very very useful. And the bioweaver means obtaining the weave off of the carrying of the expensive, and we can reduce the cost, and we can reduce the time also by this bioweaver procedure studies. So this is the next one is these are the references I used. So by this bioweaver procedures are completed.